Hello everyone, myself Hetvi Patel, and in this video we are going to discuss about regulation of signaling pathways, chemotaxis in bacteria and quorum sensing. So let's start one by one. So what is the significance of regulation of signaling pathway? Signaling pathways regulate key cellular functions such as growth, metabolism and apoptosis, and it ensures that cells respond appropriately in their environment, and it's also important for maintaining balance inside our body. And these signaling pathways are controlled through various mechanisms such as negative feedback loop, post-translation modification, for example phosphorylation, and also the synthesis and degradation of the signaling molecules. Now, the proper regulation of this pathway is very crucial because it may lead to the uncontrolled cell growth or impaired normal, which leads to ultimately the disease like cancer and neurodegenerative disorders. Now, the understanding of this regulation process is very important for us because by using that knowledge we can develop targeted therapy for particular disease like a cancer, or we can also create personalized medicine based on the individual signaling dysfunction. So now let's start with the regulation of GPCR related pathway. So as we all know that this signaling molecule comes and bind with this GPCR, and after that this activated GPCR activate G protein and start the signaling pathway. So how inside body this GPCR get turned off? So there is a GRK, G protein coupled receptor kinase is present. It came and phosphorylate this GPCR. After the phosphorylation, now this GPCR cannot work with the G protein. In this way, it reduces the signal. And on top of that, this arrestin protein come and bind with this phosphorylated GPCR, which help in internalization of this GPCR. In this way, this GPCR goes inside the cell and now it is no longer available for this signaling molecule. But when they are present inside the cell, it cannot work with G protein, but it can start alternative signaling pathways such as MAPK and JNK. In this way, GPCR get controlled through this GRK and arrestin proteins. Now the second mechanism for regulation of this GPCR related pathway is G protein inactivation process. So here this RGS protein and GRK kinases help in inactivation of this G protein. So when this RGS protein is present, it speeds up the breakdown of GTP, and as we all know, this GTP bound with the G protein and activate them. So if it get breakdown fast, then they are no longer available for activation of this G protein. In this way, signal get terminated, and this GRK kinases also help in G protein inhibition. So here this GPCR get dimerized and create a pair of receptor, for example GABA receptor. It can start new signaling function because of which the response to specific drug can get altered. It also start crosstalk in which one receptor of the pair get activated and it change the behavior of second partner receptor, because of which it either enhance the signaling or the inhibit the signaling. So what are the key implications? So we can target RGS and GRK proteins, and it helps us to manage the condition like high blood pressure or mental health disorder. So in this way, these proteins are very potential therapeutic targets for maintaining several disease conditions. And since this GPCR can interact with each other, as we see earlier that they get dimerized, so we cannot create specific drug, but it can also allow us to create a drug that can target a multiple condition. So now let's see the regulation of Ras MEP kinase pathway. So in this table, in this column, we can see that the step of this pathway in which this different protein get activated step by step, and these are the activator which help the activation of this protein at every step. Here in this column, we can see that these are the inhibitor of the every stage of this protein. So in this way, this MEP kinase can get activated and get inhibited at various steps as per the requirement of our body. So in this way, this whole pathway is regulated by this protein. So now the third pathway is PI3 kinase pathway. So let's see its regulation. So here in this slide, we can see that this PI3K pathway can get activated with various mechanism and with various protein. So when the growth factor bind with this receptor tyrosine kinase, it can activate PI3K pathway. Also this G protein couple receptor bind with specific protein subunit and start this PI3K pathway, and some small GTPase activate this RAS protein, and this RAS protein start this PI3K activation. In this way, this pathway can activate it through various mechanisms. So let's see the negative regulation of this PI3K pathway. So in the presence of this P10, this PIP3 convert back to the PIP2. In earlier video, we have seen that in the PI3K pathway, this PIP2 convert into PIP3, and then it activate AKT, and then it activate mTOR and helping other functioning of the cell. But in the presence of this P10, this PIP3 convert back to the PIP2. In this way, this pathway can get stopped and it help in tumor suppression. And as you all know, AKT get activated in this pathway. After that, this AKT activate this mTOR C protein, and which is helpful in protein synthesis, cell growth, and when the concentration of this mTOR C1 is increased, then it give a negative feedback to this pathway and give a signal it to stop this pathway. In this way, this mTOR C helpful in feedback inhibition of this pathway, and there is also other phosphatases are present which help in inhibition of this PI3K pathway. So here when the AKT is activated, it stimulates the secretion of NF-kappa B. In the presence of this NF-kappa B, the activity of this P10 get reduced. As the activity of P10 is reduced, it starts the PI3K pathway. In this way, this pathway can get turned on and turned off as per the requirement inside our body. So what is the clinical relevance? So in the case of cancer, the loss of P10 can lead to the uncontrolled cell growth, and the proper insulin signaling occur through this PI3K pathway is very important for metabolic balance, and if any issues occur in this pathway, it can lead to the diabetes and obesity. So by understanding this regulatory mechanism, we can create proper treatment by targeting that specific condition and cure this cancer and metabolic disorders. So let's see the regulation of this JAK-STAT pathway. So here this SOX protein can get activated by cytokines, and when this SOX protein is activated, it inhibits JAK it competes with STAT, and it also degrades this JAK-STAT dimer. By this, it can inhibit this JAK-STAT signaling pathway. There is also one mechanism in which when the concentration of this STAT is increased, it also increases the expression of this SOX protein, which ultimately stops this pathway. In this way, inside our body, this JAK-STAT pathway pass the signal in controlled condition. This activated state acts as a transcription factor. So it goes and binds with a specific fragment of DNA and start the transcription of the gene. But to stop the overexpression of that gene, this PIs come. This PIs come and bind with this state, and it prevents its binding with DNA. It also causes sumolization at its activation site and block its activation. It also recruits some suppression, and ultimately it suppresses the gene expression. There is another inhibitor of this pathway which is known as protein tyrosine phosphatase. So this protein tyrosine phosphatase removes the phosphate group from this JAK state and receptor. In this way, it reverses its activation. So what is the clinical implication of this pathway? So dysregulation of this JAK state pathway is linked with various diseases such as cancer and immune disorder. So by understanding this pathway and how it gets regulated, we can develop JAK inhibitor and also other treatment to prevent this disease. So let's start the second topic of this video, which is chemotaxis in bacteria. Chemotaxis in bacteria is a very clever way for them to sense and move towards helpful chemicals, which are known as chemoattractants, and away from harmful ones, which are known as chemorepellents. And this ability is very important for them to survive and finding nutrition. The movement mechanism is run and tumble motion, and bacteria like E. coli use tail, which we known as a flagella, use it for their movement. When the flagella spin counterclockwise, they stick together and make the bacteria move forward in a straight line, and which called as a run. But when they spin clockwise, those flagella spread out and cause the bacteria to tumble and to change direction randomly. So let's see the mechanism. Repellents. As we see earlier, repellents are harmful chemicals. So bacteria have to get away from these repellents. So when the concentration of these repellents is high, it creates a chemical gradient which can get sensed by these chemoreceptors. Now this chemoreceptor activate this CHEA kinase. Now this CHEA kinase phosphorylate this CHE5. Now this CHE5, which is phosphorylated, goes and bind with flagellar motor, and it cause rotation of flagella in clockwise, because of which the movement called tumbling start, and the bacteria change its direction and get away from the harmful chemicals. But when the attractant, which are the nutrients, are present in environment, it can sense by this chemoreceptor, and it reduces the activity of CHA kinase. So now as the activity of CHA kinase is reduced, the phosphorylation of CHE5 is reduced. So they cannot bind with the flagellar motors, because of which this flagella start rotating anticlockwise and bacteria continue going forward without changing direction. So now let's summarize this. When the attractants are present, the concentration of CH5P is lower and flagella rotate anti-clockwise and the bacteria move forward without changing direction. But when repellents are present, the concentration of CH5P is higher and flagella rotate clockwise and it changes its direction. What is the clinical and ecological importance? This chemotaxis help harmful bacteria such as Helicobacter pylori move towards host tissue and assist the infection. This movement allowed bacteria to gather on surface and start formation of biofilm. It also helped bacteria to find best place to grow, such as nutrition-rich areas in soil or water. Now let's see the third topic, which is quorum sensing. Okay. The term quorum sensing describes bacterial communication phenomena that allow bacteria to communicate using signaling molecule. It helps in assessment of population density. This process helps population of bacteria to collectively regulate gene expression and also their behavior. The bacteria who use quorum sensing, they constantly produce and secrete this certain signaling molecule, which are also called as autoinducer, and this bacteria also have receptor for this autoinducer. When the autoinducer bind with this receptor, it activates the transcription of specific set of gene, which is also responsible for synthesis of this autoinducer itself. In this way, the concentration of secreted autoinducer is increased outside in environment. So as we see earlier, the concentration of autoinducer in the surrounding medium, it depends on cell population density. As the bacterial population grow, concentration of this autoinducer in surrounding increase and cause synthesis and secretion of more autoinducers. And this autoinducer only can detect it when they reach to the minimal threshold simulatory concentration, and after that it alters the gene expression. So if we simplify this, the autoinducer synthesized inside the bacteria and then it gets secreted from this bacteria, and when the concentration of this autoinducer is increased outside in the environment, it gets detected by other bacteria, and when it gets detected by other bacteria, those other bacteria also synthesize this autoinducer and secrete them. In this way, the concentration of the autoinducers start increasing as the population of bacteria is more. So now let's see the quorum sensing in gram-positive bacteria. Gram-positive bacteria use process autoinducing peptide to communicate. These autoinducers are unable to penetrate through cellular membrane, so they use two-component system to control the transcription of corresponding gene, which we will see in further slides. So here in bacteria, propeptide is synthesized. After the post-translation modification, it converts into AIP, auto-inducing peptide, and now it can get secreted with the help of ABC transporter. When the concentration of this API is increased, it can get sensed by this sensor kinase. As we see earlier, this bacteria use two-component system. So here is two component. One is sensor kinase, another one is response regulator. So now when this API is detected by sensor kinase, it causes autophosphorylation at the histidine residue of sensor kinase. After then, it also phosphorylates this response regulator at its aspartate residue. Now this activated response regulator goes and bind with the specific gene and start expression of this particular gene, which ultimately synthesizes these propeptides. So here in this table we can see that in different bacteria there are different sensor kinase and response regulator are present, which detect those signaling molecule and use quorum sensing mechanism for particular functions such as Staphylococcus aureus use this quorum sensing mechanism for virulence and biofilm formation, and this Staphylococcus pneumoniae used for competence and DNA uptake. Let's see the quorum sensing mechanism in gram-negative bacteria. Gram-negative bacteria primarily use acyl homoserine lactones (AHL) as the autoinducer. These AHL are small and hydrophobic molecule, so they can directly diffuse freely through bacterial membrane, and this AHL is synthesized inside the cell in the presence of Lux I enzyme, which use S-adenosine methionine and fatty acid intermediate as a substrate. Okay, so let's see how this mechanism discovered. The first discovery of bioluminescence was found in marine bacteria called Vibrio fischeri. These bacteria do not produce light when they are alone in the ocean. However, when they grow in a large groups, they start emitting blue-green light. So these bacteria, Vibrio fischeri, often form partnership with certain fish and squid. And when they live in the special organ known as light organ inside this fish and squid, they, they thrive in a higher number and start producing blue-green light. When the research on how this Vibrio fischeri regulate bioluminescence occurred, then they discover the bacterial quorum sensing via AHLs. Now let's see the mechanism. Okay, so as we see earlier that this Lux I enzyme synthesize this AHL. As it can directly cross the membrane, it came outside the cell, and when the concentration of this AHL is increased and it reach up to its threshold, it can get detected by other bacteria, and it goes inside the cell and start the transcription of targeted gene, which include certain specific set of gene and also this Lux I. Now this Lux I enzyme gets synthesized and it start synthesizing more amount of AHL and secrete it outside the medium. So as we summarize the earlier mechanism, when the cell density is increased, concentration of AHL is also increased, and this AHL flow back inside the cell and activate high level transcription of Luxi and other genes whose product is important for bioluminescence. In this way, in Vibrio fish cherry, it start emitting blue-green light when they are present in a group. So thank you for watching this video.